So what's CRISPR-Cas9? CRISPR-Cas9 is a new technology which enables us with pretty much amazing precision to alter the genetic code. So to change the letters in our DNA sequence in a very specific way. And we can do that, and we can therefore alter the activity of genes. We can either correct mutations, or we can actually turn genes off. And that way we can actually start to think about actually providing a therapy to genetic disease at the level of repairing the DNA. How does CRISPR-Cas9 work? So CRISPR-Cas9 as a nuclease, it's an enzyme which cuts DNA. And so what we're doing here is we're basically introducing a cut within the DNA strand. And so you can think of it as a kind of a set of molecular scissors that act at a specific DNA sequence. And that cut is repaired by the cell really rapidly. And more often than not, it just sort of stitches those two ends together. But sometimes it gets a little bit wrong, and that's enough to introduce a mutation. And that can be sufficient to basically sort of destroy a gene's activity, which can be sometimes therapeutic. But perhaps the real power of this technology is when you, at the same time as adding the DNA cut, you add a template, which is basically the information that you want to put into the DNA, and that is used to repair the break in the DNA. And in that way you can actually start to think about correcting mutations within the DNA code that cause disease. Risks for CRISPR-Cas9 is an enormous field, and part of the problem is when we talk about the nuclease, we define where it cuts just by 20 of these bits of DNA code. And that 20 string, if you like, of letters is not particularly unique within the genome. And so the problem is that it might have a site which is kind of where you want to cut, but it will actually, the CRISPR-Cas9 nuclease will be cutting elsewhere in the genome. And that's really bad news, because obviously those cuts could also be repaired and mutations could be introduced, and then you could end up with this kind of lack of specificity. The other problem with CRISPR-Cas is the fact that you've got kind of collateral damage. Sometimes you think you're just doing something, but in fact DNA breaks are enormously sort of dangerous to have in the cell, and you can get all sorts of weird recombination, rearrangements of the DNA strand that could potentially cause other diseases. So there's a lot of work at the moment looking into trying to make the tools more accurate, more precise, and just to get a better feeling for how we can actually detect some of these undesirable consequences after we use them. The reproductive field is an interesting one, because obviously we were using CRISPR-Cas9 in basic bioscience to understand what genes do and all the rest of it, and then people realized, oh well, hang on a second, we could actually use this for gene therapy to repair mutations. And it was a lot of work in sort of animal models suggested that it does work really efficiently in the fertilized zygote, in the fertilized egg. And then of course people started thinking about the idea of maybe it could be used in the fertilized human embryo, and then it could actually be used to repair genetic mutations. So take cystic fibrosis, for example, or sickle cell anemia. These are diseases which are caused by just a single nucleotide, single base change in our DNA. So maybe one way of providing a therapy for such patients is actually to repair that genetic defect in the fertilized egg. Now this gets us into a huge set of ethical debates. What right do we have to fool around with the genome of our species, basically, because of course that mutation, if corrected, is not just corrected in that patient, but it's also corrected in that patient's offspring. It's going to be an interesting area, because it does have the ability to effectively eradicate certain types of human disease.